the webinar. We'll have the webinar by Joël Araiz. There's a mistake in the name in the advertisement. But okay, Joël Araiz from University of Coimbra, and Myriam Abbasi, who works with Joël, and they will talk about from molecular data to therapeutic insight, deep learning approaches in biomedical research. So Joël will do the introduction, and Myriam will go ahead after him. And Eric Rivals, I guess he's here, will moderate the session. Eric is okay. I guess he's here. Well, anyway, he's okay. It told me it was okay. So you can start, Joël, if you want.
Okay. So good afternoon, everyone, and thank you so much for the organization for considering us for this webinar. It is our pleasure to bring the work that we are doing in Coimbra, Portugal, in our lab of artificial intelligence for biology, and to bring how the work that we are doing and the ideas that we are exploring can be aligned with the cost action. So actually, before giving the floor to Mariam, that is the one that is going to show the things that we are doing, I just want to bring one idea to all of our minds. And the idea it is that with the recent advancements in artificial intelligence, I think that there is a shift happening in the way that we do research in molecular biology and different areas of biology. And I think that the thing that is changing it is that now we have plenty of data available, and we have computational resources and algorithms that are very well established. And I think that is possible to be able to do some of the research in silico. So instead of having to be us, the researcher, to read all the literature and keeping up to date with the literature, something that has been harder and harder to get, it is now able to have computers to being able to read the literature or to access the databases and to be able to replicate hypotheses in silico. And then, and I totally agree that the wet lab and being able to validate the results of this hypothesis in the lab, it's very important. But instead of doing like in virtual screening that we run through a huge number of hypotheses, then we go to the wet lab with a very specific question to answer. And I think that the artificial intelligence and these computational algorithms will have a very important role on this and bringing this shift to all of us. And I think that the pieces that are still missing are not the algorithms or the hardware, but actually are the tools. And that is something that we want to work in collaboration with you, with the ones that are on the lab, to bring these tools to the everyday user. And I think that most of the things that Mariam it's going to present, they are going to be not the tools themselves, but the methods that could be applied to this. And so that is the main message that I want to bring to this. But without further ado, I want to give the floor to Mariam. Mariam has been a very long time collaborator in the lab in the University of Coimbra. Also now is another research center institute, but we have been collaborating in these topics. So Mariam, maybe you want to start the presentation to start your.
Good afternoon, everyone. Today I'm going to present the research that I've been done during last 3 or 4 years that is about from molecular data to therapeutic insights which is the application of the deep learning models in biomedical research. My name is Paula Alepuz, and I am a researcher at the University of Coimbra. I got my PhD at the University of Coimbra, and my research focused on the artificial intelligence applied in the drug discovery and precision medicine. Today, I'm going to talk about three main topics. The first question, and I will try to answer, is that can we accelerate the process of the drug discovery? Like in generating drugs, instead of a lot of years taking, we use it just 2 or 3 weeks. Or the second question that I'm trying to answer is that, well, what if we create a medicine that's specific for a patient human genome? And then the third question is that can we predict if a person has a disease based on the whole genome of that person? Now, let's go to the first question. Can we create a design, and can we design and create a drug in a set of decades in some days? Or I would say it like this: can we create a model, deep model, that instead of generating a text, it generates a drugs? I mean that instead of having a chat GPT, we have a drug GPT. The answer is yes, it's possible. Now, let's go back to the motivation. If you look at this pipeline, this is a pipeline of the drug discovery that has different steps. Like the first step is the identification of the binding target and potential drugs. The second step is finding the chemical improvement and ADMET assessment. And the last one is the evaluation and approval, FDA approval of drugs. What happening is that the AI can enter in all of these steps and can help this process to become much faster. So in the drug discovery challenge, that does exist, and there are three main things that we always consider when we want to talk about the motivation of this application. The first one is that this process is really expensive. Sometimes it goes more than 1 billion. The process, it takes a lot of times. It can take like until 15 years. And the last thing is that we have a big chemical space, like 10,000 compounds, that has the possibility to be used as a drug for a specific disease. But then until we narrow it down and it reach to one that if it works or if it does not work, the failure is really high. That's when the AI can come and help in the process of predicting drug-target interaction, in generating new novel compounds, in optimization of the drug properties. And one of the things that I did during these last years, it was application of all of these things. Now, let's go back. In order for a machine to learn a compound that is a drug, for example, ibuprofen in that case, we can have different formats to show a component. We can have a 3D structure, we can have a 2D structure, we can have a formula, or we can use a canonical SMILES, which is a string. We call it SMILES, Simplified Molecular Input Line Entry System. That it's like a language for the molecules, and that each atom each has an item, it has a token, it has a name, like carbon, nitrogen, hydrogen, oxygen. Each bound, it can be shown with the equal, or it can, if it's a branch, it can be with the parentheses. And a good part is that the AI model can learn the 3D structure with 3D images. It can learn the 2D structures by putting the point cloud like a graph inside of the model, and also can learn from the strings. In order to train the model, we used the strings, and the strings to be converted to 0 and 1 to be fed inside of the computer. We have to have some pre-processing steps like tokenizing, padding, and encoding. So the main model that we used, and it will be using during last years, is the deep reinforcement learning. What is deep reinforcement learning? We have in the back, we have a neural network. It was the one that existed from 1970. But of course, the precision was not high. After, I don't know, in 2017, suddenly there some threshold function, it changes and it becomes HLU, and then the precision of the model, instead of being 70, it goes to 1995. And then we have a lot of layer of the neurons. So this becomes deep learning, yeah? And then when we say deep reinforcement learning, is that after the model, the deep model, try and learn the pattern inside of the data, we can have a outside feedback loop to force the model to relearn and learn better. This is like this. We have an agent that the agent learn the pattern and the language of the molecules in the SMILES format. Then it will generate a compounds atom by atom and token by token. Then we can have another model that is here, that is down there, that can say if this compound is good or is bad, and then with the feedback loop we can go back and then we optimize the weight that is inside of the deep model to force it to generate towards the compounds that will give us a better results. Of course, for the evaluation, we need to optimize, we need to consider properties. Not any compound can be considered as a drug. A drug has a good log P, which means that it has to be solvable inside of the water. So when we take it, it goes and spread towards the body, but it shouldn't be so much solvable inside of the water. Otherwise, when you take it, it goes directly to the guts. Or it shouldn't be so much a lipophilic. It means that it shouldn't lock so much the fat inside of the body. If you take it and then it was so much like a vitamin D is locked so much like a fat, then it goes and it stays inside of the fat that some of us has near the stomach, and it doesn't go and spread inside. And besides of that, and the main important things is that each drugs, normally they have a target. They have a receptor that can be enzyme or protein that is effective to the process of the disease. So it has to go and inhibit that one or exhibit or has some working area there, something do there, yeah? And we use a parameter that we call it IC50 or EC50 or KD. That is the minimum inhibitory activation, and that shows that how much if this dosage of the drug can bind to that protein and stop it from doing or stop the disease, yeah. And so we need this is the second and main important properties. And one of the other ones is the last ones that after we try to work with this model, we found out that it's really important is the synthesizability score. What is the synthesizability score? Is that although we generate, or the AI model can generate a compound, that the compound is valid. What is valid? For example, if there is a carbon, it has 4 connections. If it's a nitrogen, it has 3, or from the isomeric or other parameters of a molecule, from the chemical point of value, it's valid. But then, if we cannot synthesize that molecule inside of the laboratory, so means that we do not want to spend 1,000 euros on something that it will not work. So this is another properties that we found out that we have considered when we are generating a new compound as a drug candidate. What we did is that in the first type of models, we used all the data molecular database that is available, like Campbell, and that has like 1,500,000. We used the drug bank, and then we use the biogenetic databases, zinc, and then we use some AI to generate novel compounds. Yeah. In order to do this, we use the SMILES in the first step, and then we use some model that I'm not going to explain in detail. What does it do? It's a recurrent neural network. Is like a model, AI model that is at the back of the Google Translate that can learn the language and then convert one to the other one. So with this model, we could have generated compounds, and we generated like 1,000 compounds, and we compared with the state of the art, and we saw, okay, our model is good. But that's not the point. With the things that I really wanted to show you is that although this is AI, but there are a lot of parameters, we call it hyperparameters, that when we are trying to train the model, we have to optimize them so we can get a higher precision. So, and right now I explain just this part, the part that we are generating the compounds, but we do not want just to generate the compounds. As I said, we want to have a reinforcement learning. we want, after we generate a batch of smiles or batch of drug candidate, we have another model that model can predict the binding affinity. For example, in this case was the adenosine, and then based on the value that if they are good or if they are not bad, we give a reward to the generator to generate compounds according to what we want. And this work also, of course, is published in the Journal of Bioinformatics, and then it already has 150 citations. You see here we have two generators. We have the unbiased and we have the biased generator. The unbiased one is the one that it works like aleatory. It generates like randomly. So and the one that is biased is always being updated. They always update the weights according to the generated compounds. So this one gives diversity to the generated molecules, and this one make it fine-tuned towards the correct compound. As we can see, the result, it shows that the molecules that we generated, they could have a good diversity and they have a high binding affinity. Of course, as I said, a compound cannot just have a good binding affinity. For example, if it's for the Alzheimer, and so it has to go to the brain and it works there. So it has to pass through the blood-brain barrier, and then blood-brain barrier are so thin, so not everything can pass. So in that case, we adapted our model to do not just create compounds that can pass, that can have a good binding affinity. They also can pass through the blood-brain barrier. And then with some algorithm, and that is multi-objective optimization, we join these two reward and then try to force the generator, the unbiased generator, to generate a compound that have both properties at the same time. So this is another model. This model also does the same. It's not the reinforcement learning. It's like evolutionary algorithm with the GANs. I just wanted to show to you that it's not just one thing that does the job. There are a lot of different structure that they can learn. For example, this GAN or generative adversarial network are the deep models that is generated based on how the human being brain works. For example, in our brain also we have one side that is generate, and then we call it generator, and we have another side that is the critic, that try to predict if the things that's generated is fake or it's real. So we use the same structure here. It's a little bit more complicated because this model cannot get the smiles in the format of a string. So we have to convert the smiles in some feature latent vector, and then we feed that one to the model and then try to optimize it. And of course, this model also works well, and it was good enough. For the study limitation, I would say that although the models, they are working and they give like 95% accuracy, but then we always have the problem of the limited size of the data to train, like for example, if it's for the target, a specific target. And also another main important things to say that is explainability. For example, if the model is saying this is good, why it is good? Maybe it's not real good because we never try to synthesize and test in the laboratory to see, yeah, is it really good or it's not. Now, let's go to the next things. I think this part is more interesting for this group, because in this part, what we did is that we focus on translation regulatory genes. We try to design a drugs for the cancer, but we didn't use the information of the target because we didn't know which target we want to consider. We just used the gene expression data of the cancer people. So in this one, what we did is that we use the computational agent, we use a data set to train, but then we use the transcriptomic data to help this process. To do this, what we did is that since we have transcriptomic information, we download this data from the drug database TCGA, and then we use some variation algorithm encoder. That doesn't matter. It's like a deep model that gets table data and it converts it to the feature vector. It's a vector that's smaller, but it has all the information that is existing inside of this one in a compact version. So here we have exact amount of data here, but in a smaller version. So it can be used in the deep model. And then this part is exactly was the one that I was explaining. Now we get a generator, smiles, we convert it with the variation encoder to another vector. Then we combine these two vectors that they are not really related to each other. One of them has the information of the gene expression data, and the other one is the SMILES information, a drug. With some fusion model, we convert them, we join them, and then we try to train the model so it can so the drugs that we generate, it can vice versa the gene expression information in such a way that if a person, because we are training with the cancer data, for if the gene is expressed high, or so we wanted to do the contrary, or if it's expressed less, we wanted to express more. So we use these CDs with the function that we call the reverse gene expression scores, and we combine these two, and we use again the reinforcement learning to force the model to generate. Of course, this work also published in the Preference in Bioinformatics, and it gets a lot of notice inside of the news of Portugal, because it was one of the first model that is trying to create cancer drugs without information from any type of a specific cancer, just from the gene expression data of the cancerous people. So this was the result. It doesn't matter what it is, but it shows that, for example, for 10,000 compounds that we generate, we just pick up the one that was here, that is maximizing the both two properties that we wanted to consider. And if you see, after trying to create the scaffold of these molecules, we saw that the model can generate compounds that never saw, and then the some parts of the scaffold are similar to the already cancer drug that does existed in the market. So this means the model could learn how a drug should be used for the cancer without having any information about the target or about the type of the cancer. Just with having the gene expression information, it could have trained and generate molecules that can be used as a drug candidates. So of course, this also has some lack of some limitation, like sufficient training data, how gene expression profile is computed, and then to train the model for the gene expression data, we didn't use all the genes. We used the landmark gene size, which is just 1,000 that we found from some other works that they said that they are the most considered one that might be interested for the cancers, but these are our limitation of this work. Now, this part is about that. Until now, we always trying to create a drug that can be used for a therapeutic ideas, like for the cancer, for Alzheimer, or any kind of disease. But then when we went to the laboratory and then we asked some collaborator to try to synthesize it, we saw that it's not possible, because even after we synthesize it, and then they tested in the laboratory, they did not penetrate inside of the cancer cells. So we tried to change the model. Instead of generating drugs, generate peptides. And then now, because the time was past and we are already in 2025, we tried to create or generate peptides. What is peptides? Peptides are a small fragment protein. But the good part of this protein is that since they are small, they are like a maximum 20 that we generated it, because they can degrade fast. They are liquid, and because they are piece of the protein, they do not have toxicity like the drugs. And the other part that they are really good, they are naturally existed inside of some fruits and yogurts, like that probiotic yogurts that we use. And also we can test it easily in the lab after we generated. So in this case, what we did is that we generate some peptides that is antimicrobial, because of the problem of the antibiotics and then all the drugs and that they are not effective. So it would be something that it can kill the bacteria and it does not have toxicity. And since it's just a bacteria, you can test it easily inside of the lab. In this case, we use the transformer. The transformer is a kind of deep model that is used in the back of the large language models like Gemini or ChatGPT, and we use it when we use it also the same for the evaluator to see if the peptide it has the capacity of the antimicrobial activity or it does not have toxicity. And then we join these things and then we try to train again the model and then to generate. What is the good news? The good news was that after we try and we use these deep models and we generate the peptides, we found these compounds or these peptides that I put them in the star because we are trying to make them patents, that they show good results for the E. coli in some other microbes, and they did not have toxicity. I mean that it was the only point that we reached to the laboratory, to the real, and then it was a good news for us. Of course, this also has some limitation, like the quality and diversity of the training molecular data sets and the complexity of the network and the data. Now, let's go to the second questions. This part is much easier and is faster. So this work was done with other students of the lab. So in this case, what we were thinking at the start is that what if we have something, a drug that can be specific for each person? The main idea comes from here. In current medicine, what we have is that we have a drug that we prescribe it for everyone. For some people, they are effective. For some, they don't have effect. For others, they have adverse effect. For example, if my son has ADHD, I give him the Hobifen and then it becomes like numb. He doesn't talk, he doesn't eat or anything, but in some others it works. What if we create in the future a medicine for each group of people? So it's effective for all. I mean that we could generate one for the first group, one for the second group, one for the last one that has problems. And then like this, we always have effective drugs. So it was a really interesting idea, and I think it's really nice. It's really good. If we could have done it. But after studying a lot of papers, we found out that this is not possible because we do not have enough data and we do not have enough information up until now, and it's really hard to create and generate a model to do that without any kind of data. So what we did is that we used again the gene expression data from the TCGA because the gene expression data of the cancer people are available. We use it for different formats. That's not important. We used again smiles in the format of the string. That's also it was okay. We used the autoencoder that converts this information to a feature vector. But in this case, what we did is that if we can create a model that does not generate, but just can predict if a drug is working for a specific diseased person or not. And yes, it was possible. Although we just have 256 drugs, and we test just 256 drugs that works for the cancer, and we have just the information of the cancer people that was 12,000 and mutational data, we could have predicted this like 85% accuracy in that if the drug is effective for that person or not. And also we use other models like the convolutional neural networks, that the one that works for the genome to see if it works. And yes, it did work. So what is the conclusion is that we can create a model that can predict if a drug is effective for a genome of a specific person, but still we cannot generate it because we do not have enough information. Now, most and the least, let's go to the last question. The last question also we did with some other students and some other groups. We wanted to see if we can predict the person has a specific disease or not. And yes, we could. In this case, we use the information of the schizophrenia from some Swedish to predict if that person has schizophrenia or it does not have. As you know, we cannot do this for all kinds of disease because some of them, they have a lot of parameters like environment, I don't know, a lot of things that might be effective, a mixture of a lot of things that lead to a disease. But in the case of schizophrenia, that the reason is completely genetics and comes from the parents, it was possible. We created a model and we trained it with the schizophrenia data sets and that we got it from some laboratory from the Swedish, and we trained and we generated a model, deep model that has it has a lot of things, and then we could predict just based on the genome, without gene expression and without any other things, to see if that person has a schizophrenia, and it does not have, with 80%, 85% accuracy. Here are some of our publications, and just some of them that might not be interesting. And this is our group. Some of the students are not with us, but the work was done with them. This is the link of GitHub. All the codes of all the works that they have done that I did not even present are existed there. Thank you so much.
I'm not
Is
sure
there any
if there
question?
are some questions or
Yeah.
things.
Yeah, we can start the question time now. If you
Okay.
have a question, go ahead.
I have a
Carmen.
quick one, Marian.
Okay, sure.
So in your models, yeah, for example, at the beginning when you were picking like small molecules, and when you were looking for a target, so you needed to know the target, like the protein target,
Yeah.
right? So that's how this system
Yeah.
works.
Yeah, exactly.
Okay, all right. So is let's say is based on targeting the same place, or let's imagine the pocket on a protein that it is already been targeted before, more or less.
Yeah.
Okay.
Yep.
All right.
It's it's like this. In order to evaluate if a compound will be bind to the target or not, we had to download the data from the Campbell, all the compound that does exist that is already do this connection, does have this binding affinity to train the evaluator. So it's not for finding the target. It's using the target that is already known and then generate compounds that can bind to that specific target.
Can I just make a quick comment? So
Yeah, sure.
everything is correct. So the easiest way it is to optimize the target, the function for a well-defined target. But in one of the works that Mariam presented, we can, instead of guiding the function for a specific target, we can guide it for the transcriptomic response. So if we do not know the target, we can guide it to a given transcriptomic response. And so implicitly, we are going to discover the target, but that in that specific case, it's not something that we are giving in the beginning. And I think that this is the most interesting part of these models, is that we can change them, the structure, to the proper properties that are of more interest for us for a given problem. So yeah, so in this one specifically for the TCGA, we didn't know that. So actually, we knew the target, the USP7, but we had no prior information to the model about the target, and only and based on the transcriptomic response that we wanted, we were able to identify that the target that we wanted, it's actually the one.
Yeah. Thanks.
Also, we had another model that I did not show it here, that it does not use the specific targets, but it can evaluate and train the model to find the specific parts of the functional groups of each compound based on the attention heads of the
transformer model.
Yeah. All right. Cool.
More questions?
I have a quick one, not really with the models, but at the end you said that your model can predict schizophrenia.
Yeah, yep.
I was wondering if it's predicting, that means it's before the people is sick.
Yeah,
So
yep.
that-
Exactly, it's like that.
Okay, so does that mean that it can have some prevention not to become schizophrenia, or
Yeah,
yeah, it could be?
Yeah, it could be, because what does the model is that it does not use the environment information, it does not use the gene expression data, so it's not it's not inside of the body yet. Yeah, we use the whole genome that is like that one that has the LLA information like 1, 0, 2, yeah, and then we compare it with the human genome data, the original one, and then based on that we just use that one. Then we saw that okay, the genome is really big, yeah. So we with that part of the parts that we could have used, we did it like this. Use some models that connect the three of them that is inside of the genome, and based on that we found. Okay, we found out that which one is. Then we use the AI explainable. Yeah, that's what that is interesting part that I did not have it yet. To find out, after it's predicting if a person has schizophrenia or not, based on, for example, just they took out your blood and they sequenced your genome, and then we give it to the model, and the model will say, okay, you have 85 with the probability of 85%. You have a schizophrenia, yeah, and then after that we can say exactly based on which genome inside of your body you have this, because we use the explainability to go backwards and use to see what causes the model to predict that this person has a schizophrenia. And then we compare it with the state of the art, the genomes that is already, they said it that is related to schizophrenia, and they were like more or less like 70% was as the same.
Okay.
With enrich, because this part it was with Daniel, and he has more information about the computational biology. He did the enrichment analysis, and then he found out with other professor that we are collaborating. So it for that part was really interesting part of the work. We say that AI responsible or is a black box. No, it was not really black box. We trace it backwards, and we found that the exact part that I think is the interesting part.
Okay.
Thank you, Eric.
So if I'm not sure if there's other questions, but just a final, is there?
Eric, do you want to
Eric.
ask something? If you want, we cannot hear you.
Yeah, maybe you can put a question in the chat.
Eric, can you hear us? I don't know. There's a problem.
Yeah, he can.
Okay. And you wanted to speak, or
Because we cannot hear you, Eric, at all. We see your hand. You're like, pick me.
Some more person like problem.
okay? Somebody else some question?
Maybe Eric wants to write it down. Oh, he wrote it. What about the bias in database regarding the distribution of species?
Oh, okay. Which, this is something that we did not try to answer, or we did not consider it. But we try to filter data as much as possible and use the sample after, because the data set was like 1,200. But then we cut it and we use the one that is really has the significant genomes. But regarding this, we didn't do any investigation.
A quick question: how much computer power you need to do this?
Short answer, not enough.
Not enough. Okay. All right. Thanks. That tells.
So yeah, so we are always trying to improve our computational infrastructure, and from now on we are relying on the data center from the research institution that is being built, but it is a race against the obsolescence of previous hardware with 3 or 4 years old.
Yeah. All right. Eric has continued his question, and he said, I meant it is usually biased toward species that we can grow and manipulate in the lab. I guess he's also talking about bacteria in the case of.
Oh, you are talking about bacteria. I think that he is right. Genetic prediction models, or I was thinking it's about the genetics. They are usually or historically, they are biased towards the species and the population that they are easy to grow. Yeah, this is a real, well-known limitation. But we did not ignore it, but we explicitly modeling, and we are trying to correct it. Yeah, of course, he's right. But then there is something else about the computational. We had another student that he's trying to implement a model that just predicts the generate a new compounds, not with the smalls, in the 3D structure with the molecule in the format of 3D structure. So, it has to train a 3D version of the molecules, and then we were trying a lot, and then we found out that our computer in the lab, it really does not work, because in order to training it takes like 21 days or something. Yeah, of course this part is hard. That's why we use the smiles. That limitation does exist.
Well, also, I want to complement that a lot of the things that were presented today by Mariam, they are, like the technical nuts and bolts of the things that we are doing in the lab. But I think that it is also clear that we are quite open to apply this to other problems and we are quite open to further collaborations if you see that there's the potential of the methods that we do to get novel results. And I think that this is the most important take-home message.
Yeah, exactly. It's like this, since we are informatics, we know how to work with the data. It doesn't matter what it is, as long as there is data, we can use different type of models, as you can see, that does exist, and they are improving every day to be applied. And it's not just applying or predicting or generating, it can also get conclusions or use like explainability to see why it is like this.
Okay, so is there more questions around?
So, thank you so much.
So yeah, I think maybe we can stop here if there is no more question. Thank you very much, both of you, Mariam and Joel, to give us this presentation today. And I hope maybe somebody here that they can collaborate with you if they want. So maybe it will happen. Okay, for me, that's it.